Continuous culture. Continuous culture is one of the cultural process in which the bacterial population grow at a constant cell concentration and volume within the culture vessel or reservoir. It is a type of open cultivation system. There will be the continuous addition of the nutrient medium and the constant elution of the product. In the continuous culture, the growth rate of microbial cell is maintained in the exponential phase, that is, the cells could not reach the stationary phase of growth. The process of maintaining the microbial cells in the log or exponential phase refers to the steady state of growth. Apparatus for continuous culture. One requires some additional equipment to maintain the microbial cell steady phase to perform the continuous culture method. Chemostat and turbidostat are two unique types of equipment which are most commonly used in continuous culture. Chemostat. It is an apparatus used for the continuous culture of bacteria where the cell density is kept constant by keeping the dilution rate and the flow rate of nutrient medium constant. Chemostat was introduced in the 1950s by three scientists Monod, Novick and Szilard. The sterile nutrient medium is added into the reservoir along with the microbial cells. The chemostat depends upon two factors like dilution rate, the concentration of limiting nutrient. The change in dilution rate will change the growth rate of bacteria, that is both cell growth and cell density. By limiting the concentration of an essential nutrient, one can determine the nutrient medium's flow rate into reservoir. Therefore, this essential nutrient will also determine the bacteria's growth rate. Culture of bacteria by chemostat. To perform the continuous culture by chemostat, there are three elements like substrate reservoir, culture vessel, and the spent culture bottle. The sterile nutrient medium is continuously added into the culture vessel via flow regulator. The flow regulator allows the constant flow of the fresh substrate into culture vessel. There is an inoculation pod in the culture vessel from which the inoculum of bacteria is added. The culture vessel compresses the magnetic stirrer at bottom, allowing uniform mixing of cells within the nutrient medium. The remaining dead cells and byproducts from the culture vessel are released from the sampling outlet into spent culture bottle. Turbidostat. Turbidostat is a device that maintains the constant cell density by controlling the flow rate of fresh medium. It is also called biostat. It is introduced by two scientists, Bryson and Przewalski, in the year 1952. Its working depends upon two components like photoelectric device, optical sensing device. A photoelectric device like photodiode performs a key role in examining the cell density in the culture vessel. An optical sensing device measures the turbidity or absorbance of culture in the reservoir. Culture of bacteria by turbidostat. A turbidostat is an apparatus which includes a sterile reservoir, culture vessel, photocell, and a light source. The fresh nutrient medium is added to the culture vessel through a valve which controls the flow of medium, so the optical density increases. The cell density is kept constant in the culture vessel by help of photoelectric device. The turbidity is monitored throughout the process. Application of continuous culture. It is used for the production of citric acid in industries. Used for continuous manufacturing of recombinant proteins. It have been applied to basic physiology study of wild type cells. Chemostats are frequently used in the industrial manufacturing of ethanol, used in the biotechnological industry as an experimental model. Advantage of continuous culture. The process is cost effective. It reduces the number of stages like cleaning, sterilization, prepare of starter culture, etc. In continuous culture, the cell population can be maintained in the exponential phase at constant cell concentration for a long time. By reducing the number of stages, it also saves time and energy. It avoids the accumulation of toxic substance. It gives high productivity by utilizing the substrate. Disadvantages of continuous culture. Not as efficient as fed batch cultures in the production of secondary metabolites and recombinant proteins. There is a high risk of contamination as the process continues without cleaning and proper sterilization. High technical skills are required to maintain the bacterial cell steady state. It requires some additional and specific equipment. Thank you.